Thank you, everyone, and welcome back to the ACIP meeting. We'll reconvene now. Our first presentation for the next session will be from, I want to make sure I get the, oh, thank you, from Dr. Miller, who will be speaking about booster doses of Moderna COVID-19 vaccines in adults, adolescents, and children. Dr. Miller.
Yes, thank you so much, Dr. Lee. Good afternoon. My name is Jacqueline Miller, and I'm the therapeutic area head for infectious diseases at Moderna. Thank you to the CDC and the ACIP for all of your hard work and excellent guidance through this pandemic. It's my pleasure this afternoon to present the clinical data for Moderna's Omicron-containing booster vaccines. Next slide, please. Thank you. So for the fall U.S. booster campaign, Moderna is producing a 50 microgram bivalent vaccine containing 25 micrograms of mRNA encoding the original strain spike protein sequence and 25 micrograms encoding the sequence for the spike protein of BA.4/5. The indication is for a single 50 microgram booster administered at least 2 months after either completion of the primary series or receipt of a booster dose of any authorized or approved monovalent COVID-19 vaccine. Next slide. Although mRNA 1273 protects against variants of concern, and especially in the case of severe disease, the purpose of adding mRNA encoding variant sequences to the booster doses is to improve the immunogenicity against Omicron lineages, which have remained dominant for the past 8 months, induce a broader cross-neutralization response to other potential variants of concern, and to extend the duration of protection. In the slides that follow, I will share the data that consistently show the ability of these bivalent variant vaccines to address these goals. Next slide, please. Slide. Thank you. As the virus has continued to evolve, Moderna has evaluated 3 monovalent and 4 bivalent vaccine candidates designed to address variants of concern. More than 7,000 adults have received a booster dose of one of these vaccine candidates. Clinical data are primarily available for the Beta and Omicron BA1 vaccine. The BA.4/5 bivalent vaccine was in a clinical trial which completed enrollment last week, and data are anticipated to be available at the end of the year. Just a note about nomenclature. You'll see 3 candidates mentioned in this presentation, which combine 25 micrograms of mRNA encoding the original strain and 25 micrograms of a variant of concern. The 211 vaccine contains the Beta variant. The 214 vaccine contains BA1 Omicron, and the 222 vaccine contains BA.4/5. To avoid confusion about the numbering system, I'll be referring to these bivalent variants by the name of the variant of concern sequence that's contained in the vaccine versus mRNA 1273. Next slide, please. So I'd now like to review the available clinical data for the bivalent SARS-CoV-2 vaccines targeting variants of concern. Next slide. Currently available clinical data includes safety, immunogenicity, and 6-month antibody persistence data from 300 participants who received the Beta-containing bivalent vaccine. Median follow-up in these subjects is 245 days after the 3rd dose booster. After the 4th dose, safety and immunogenicity data are available in 437 participants who received the Omicron BA1 containing vaccine. And these individuals have been followed for a median of 43 days. These clinical data form the basis of our submission for bivalent boosters. There are also pending data from the 512 participants who were recently vaccinated with the Omicron BA.4/5 containing vaccine. So this trial completed enrollment last week, and the data are expected later this year. Next slide, please. Slide. The demographic data from our study of the BA.1 bivalent vaccine are presented here as compared to the mRNA-1273 fourth dose control group. Third doses in these groups were given approximately 8 months after completion of the primary series, and then the fourth dose was administered approximately 4.5 months after the third. About 1/4 of these participants had evidence of prior SARS-CoV-2 infection at the pre-booster time point. Next slide. So on this slide, I am displaying for you in the darkest blue the fourth dose BA.1 Omicron-containing bivalent booster compared to, in the lightest blue, post-dose 2 with mRNA-1273, and in the medium blue, post-dose 3 mRNA-1273. In this study, the solicited local adverse reactions included injection site pain, erythema, and swelling, as well as axillary swelling and tenderness. And the reported rates after dose 2, 3, and 4 are at least similar or lower than mRNA-1273 second and third doses in the bivalent Omicron BA.1 containing fourth dose vaccine. Importantly, there were no grade 4 events reported after the BA.1 booster. Next slide. And now this slide shows the same 3 groups in terms of the systemic solicited symptoms. And in this case and in the previous case, the hashed bar represents the grade 3 events. Overall, both the frequency and severity of the solicited systemic reactions were observed to be lower after a bivalent fourth dose booster than after mRNA-1273 administered as a second or third dose. And once again, no grade 4 reactions were reported after the BA.1 containing booster. Next slide. Now this slide reviews the unsolicited AEs that were reported within 28 days after vaccination. Rates of each category of AE were similar between the mRNA-1273 and BA.1 bivalent vaccine groups. Among the 3 SAEs reported, cases of prostate cancer and traumatic fracture were reported in the BA.1 bivalent group, and a single case of spinal osteoarthritis in the mRNA-1273 group. None of these SAEs were considered by the investigators to be vaccine-related, and there were no cases of myo or pericarditis reported in either group. Next slide, please. So now we'll shift to talking about the immunogenicity analyses, which were used in this study to infer effectiveness. The primary objectives of our study were to demonstrate the superiority of the BA.1 containing vaccine over 1273 in terms of neutralizing antibody responses to BA.1, and to demonstrate the non-inferiority of the bivalent vaccine to 1273 in terms of the original strain neutralizing antibody titers. So this slide first shows the BA.1 superiority hypotheses. There were 2 criterion to assess success. The first was that the BA.1 GMT ratio had to have a 97.5% confidence interval lower bound, which exceeded 1.0. The actual ratio was 1.75 with a lower bound of 1.49, so this criterion was met. The second criterion was that the lower bound of the 97.5 confidence interval around the group difference in seroresponse rate would exceed -10%. And this was also met with a point estimate of 1.5% and a lower bound of -1.1%. Slide. So now in terms of the non-inferiority hypotheses to the immune responses against the original strain. In this case, the lower bound of the 97.5% confidence interval had to be greater than 0.67, and this was met with a point estimate of 1.22 and a lower bound of 0.108, or 1.08, excuse me. For the group difference in serial response rate, the lower bound had to be greater than -10%, and this was also met as the response rates in both groups were 100%. And therefore, the study met the primary objective to demonstrate superiority in terms of BA1 antibody titers and non-inferiority in terms of mRNA, in terms of original strain neutralizing antibody titers. So next slide, please. So importantly, the primary hypotheses were evaluated in subjects who had no evidence of prior infection. But we also know that at this moment in time, many vaccinated individuals have experienced SARS-CoV-2 infection. And as I mentioned, it was about a quarter of the participants in this particular study. So this slide shows the geometric mean titers and the fold rises to BA1 in all participants, regardless of previous SARS-CoV-2 infection. And you see it both for the mRNA 1273 group in medium blue and the Omicron BA.1 containing bivalent vaccine in dark blue. So even in subjects with evidence of prior infection who were observed to have substantially higher pre-vaccination titers, derived benefit from the BA1 bivalent vaccine, with a four-fold rise in post-vaccination titers to Omicron BA1. This exceeds the 2.5-fold rise observed in the mRNA 1273 group and led to a GMT ratio of 1.9 and a lower bound of the 95% confidence interval of 1.5. Next slide. Improved antibody titers to the Omicron sublineages was a major objective for developing the bivalent vaccine. And this slide shows the consistency of that finding across age groups. So you see the antibody titers to BA.1 and the original strain stratified by age group, 18 to less than 65 years and greater than or equal to 65 years of age. Post-vaccination, the BA1 bivalent vaccine was consistently immunogenic to both the original and BA1 strains in subjects over 65 years of age who are at increased risk for the severe complications of COVID-19. The observed antibody titers against the BA1 variant of concern were higher with the bivalent vaccine as compared to 1273, regardless of age stratum. Next slide, please. So we also examined antibody titers to the original and BA1 strains in subjects stratified by race. For both strains evaluated, the BA1 bivalent vaccine was consistently immunogenic across age groups. Next slide. The induction of cross-protection to other Omicron sublineages was another major objective for the development of the bivalent booster vaccine. We evaluated the ability of antibody titers induced by BA1 bivalent vaccine and mRNA 1273 to cross-neutralize BA.4/5, stratified by evidence of prior infection. Both vaccines demonstrated increase in GMTs to BA.4/5, which showed evidence of cross-neutralization, but the higher antibody titers induced with the BA1 booster give us reason to anticipate that the BA.4/5 bivalent vaccine, which is matched to the currently circulating strain, will lead to improved antibody titers. Next slide, please. We have also stratified the antibody titers to BA.4/5 by evidence of prior infection and age group. And as observed with the BA1 antibodies, the bivalent vaccines consistently induce numerically higher titers against BA.4/5 across age strata, which indicates that those adults over 65 years of age will also benefit from cross-protection conferred by the bivalent vaccine. slide, please. Bivalent vaccines were also developed to induce better cross-protection to strains which are not included in the vaccine. On this slide, we show antibody titers and relative GMT ratios in terms of binding antibodies for the BA1 bivalent vaccine compared to mRNA 1273 at 28 days post-vaccination, and for all variant studies, including alpha, beta, delta, and gamma, binding antibody titers were significantly higher with the BA1 bivalent vaccine as compared to 1273. Recognizing that neutralizing antibody data are also important with this vaccine, we are in the process of generating those data and we'll share them when available. Next slide, please. So I also wanted to show you data with respect to antibody persistence, because this is another important consideration for the bivalent vaccine. These are data from the individuals who received the beta-containing variant vaccine. And what you see in each bar is the geometric mean ratio compared to mRNA 1273. This is comparing a third dose to a third dose at first day 29 and then day 181, or 6 months after vaccination. And it's showing it for the original strain with D614G, and also the beta, Omicron, and Delta variants. And while the GMT ratio for the beta is consistently higher than 1, importantly, the GMT ratios increased even further for the original strains, beta and Omicron BA1 at 6 months after vaccination, which suggests that the bivalent boosters may improve the longer-term cross-neutralizing antibody durability. Next slide, please. So now moving to specific data which have been generated with the BA.4/5 bivalent vaccine, I would like to present the non-clinical data in mice, which has supported our EUA submission. Next slide. So we evaluated the BA.4/5 bivalent vaccine in a murine animal model expressing a human ACE2 receptor. These mice were previously primed with mRNA 1273 and then were boosted approximately 31 weeks later with the BA.4/5 containing bivalent vaccine in purple, the BA1 bivalent vaccine in orange, and mRNA 1273 in blue, with a sham vaccination presented in red. The two bivalent vaccines both induced statistically significantly higher antibody titers against the Omicron sublineages as compared to mRNA 1273. Next slide. So these mice were then challenged with 10 to the 4th plaque-forming units of a BA.5 strain 4 weeks after the booster dose was given. And these graphs show the number of SARS-CoV-2 viral copies obtained from the lungs and the nasal turbinate. The two bivalent vaccines reduced copy numbers in challenged animals, particularly in samples taken from the lungs, which is an important marker of severe disease. And as we have consistently observed that results from our murine challenge studies correlated with observations in human clinical trials, these data are supportive of the immunogenicity of the BA.4/5 containing bivalent vaccine. Slide. And finally, I will mention that we are generating data with booster doses in the pediatric population. Next slide. And then one more slide, please. Our submissions of booster data generated in children 12 to 17 and 6 to 11 years of age are currently ongoing to the US FDA. The original pediatric studies were extended to evaluate booster studies of mRNA 1273 in these populations. And similar to adults, where the booster is administered at half the dose of the primary series, adolescents receive a booster of 50 micrograms after a 100 microgram primary series, and children 6 to 11 years of age received a 25 microgram booster after a primary series of 50 micrograms. We expect to complete these submissions by mid-September. Next slide. And then in the youngest age stratum of children 6 months to 5 years of age, we're evaluating a primary series with the bivalent BA1 containing vaccine and booster doses with both mRNA 1273 and the BA1 bivalent vaccine. Results from this trial should be available by the end of the year. And we are also exploring ways to evaluate primary series and boosters with the BA45 vaccine. Slide. In summary, the bivalent booster vaccines have been generally well tolerated in individuals over 18 years of age. The local and systemic reactogenicity are generally similar to or lower than that observed with dose 2 and dose 3 of mRNA 1273. No new safety concerns have been observed in our booster vaccine studies. Our pre-specified immunogenicity objectives, which align with FDA guidance for the licensure of bivalent booster vaccines, were met for superiority to BA1 and non-inferiority to the original strain. There were significantly higher antibody titers to BA45 with the BA1 bivalent vaccine, and binding antibody titers were also higher than 1273 against more distant variants of concern. The higher immune responses were also observed in individuals over 65 years of age who are at the highest risk for the complications of severe COVID-19 disease. Our beta-containing bivalent vaccines demonstrated improved durability of neutralizing antibodies compared to mRNA 1273 at 6 months after vaccination. Preclinical data for the BA45 containing bivalent vaccine in mice is supportive of vaccine effectiveness, and this is being verified in an ongoing clinical trial. Next slide, please. So I'd like to conclude again with reiterating our thanks to the ACIP and for the opportunity to present these data. And thanks once again to the investigators, site personnel, and study participants in our clinical trial. Before taking any questions, there was a question stated earlier in the discussion around the presentation of booster vaccines. And so I'd like to say that the bivalent BA45 vaccine will be presented in a 2.5 ml vial. It's intended to have a 0.5 ml administration to adults over 18 years of age. And as we're reducing the number of doses in the multi-dose vials, we're working towards the future looking for single syringe presentation. I'll conclude there and ask if there are any questions I can answer.
Thank you very much, Dr. Miller. This presentation is now open for questions. Dr. Brooks.
Yes, thank you very much for the presentation. You showed a slide that had viral particles in the respiratory, upper respiratory, and then in the lungs of the mice. In your clinical studies for the other vaccine, I mean for the other age groups and with not booster, did you measure viral particles in the respiratory tract? If we're going to look at that kind of as a correlate of vaccine efficacy in adults?
Yeah, so I'm going to call in a moment on one of my colleagues from the research group who actually conducted these studies. But what I will say is we did conduct challenge studies in the mouse model prior to initiating our first clinical trials with mRNA 1273. And we did so to give evidence that the vaccine is capable of inducing antibodies that can neutralize virus and protect against infection in the various areas of the respiratory tract. So ultimately, the best data, of course, come from human clinical trials. As we switched from the BA1 variant vaccine that we had initially prepared to discuss at the VRPAC in June over the summer to the BA.4/5 containing vaccine, we have been able to manufacture doses and conduct the clinical trials in the mice more quickly than we've been able to execute the human clinical trial. So the data then further supporting the BA.4/5 will come from the clinical trial whose enrollment just completed, and that will present later this year. But the Beta-containing variant and the BA.1-containing variant vaccine both performed in a similar way, demonstrated superior antibody titers to the variant of concern as compared to 1273 and non-inferiority to the original strain. So we believe that the consistency of the clinical results are really the basis on which we are licensing or authorizing the vaccine. But I'm going to ask Dr. Edwards if you want to make any other comment about how those challenge studies were done.
Yes, thank you, Dr. Miller. My name is Darren Edwards, and I've led the research for our SARS-CoV-2 vaccines for the last 2.5 years. What I can say about the animal studies that we performed over the course of the development of our original vaccine, as well as in the evaluation of our bivalent vaccines, is what was represented as a common measurement that we perform in animal studies. And that includes both mouse studies, hamster studies, as well as non-human primate studies. It's a good measurement to indicate the level of protection that's provided for the different vaccine regimens. Unfortunately, it's not something that we can perform clinically due to constraints around certain types of sampling that we could perform within clinical studies. So that's why we've either relied on efficacy measurements or post-clinical observations in order to establish efficacy. Thank you.
Thank you.
Thank you. Next, we'll move to Dr. Aes.
Thank you, Dr. Miller, for that presentation. I have a question about whether we have any data on breakthrough cases, clinical cases in the BA.1 group versus the mRNA 1273, or whether there has not been enough follow-up time for that data.
So we don't have those data yet for the BA.4/5 containing vaccine, but because we have the median of 43 days of follow-up after BA.1, we do have those data. And there's disease incidence rates in the 1273 group versus the BA.1 containing vaccine group were followed and compared. They were followed both for subjects with no evidence of prior infection and subjects with evidence of prior infection. In the BA.1 bivalent group, the incidence rate was 2.5%, and in the mRNA 1273 group, the rate was 2.4%.
Thank you.
Thank you. Ms. Bata?
Thank you, and thank you, Dr. Miller, for this succinct overview. I really appreciate it. I'm wondering if we could go to slide 18, and if you could talk a little bit, you talked about significantly higher, and to me it looks like the confidence intervals are overlapping. So can you explain this to me?
Yeah, so the geometric mean ratios, which are given in the top bar, that's immediately below the name of the variant of concern, is followed by a 95% confidence interval. And those 95% confidence intervals are all above the value of 1. And so these are the between group comparisons that we perform in an exploratory fashion to look at comparisons between groups. They do, to your point, need to be interpreted a bit conservatively because the multiplicity adjustments are really made for the primary endpoints of the trial. But we do feel that these data are really indicative that we may expect to see higher neutralizing antibody titers, as we've seen for the beta-containing vaccine at 28 days after vaccination, and then obviously larger and tighter, higher GMRs and tighter confidence intervals after 6 months.
Thank you.
Thank you. Dr. Sanchez?
Thank you for that presentation, and certainly we need to move forward with these vaccines to cover other variants. But I do have the studies that you're planning to do, or that you're ongoing in children 6 months to 5 years, are they also, and I think this is slide 25, and you may have said this, but I may have missed it, these are also with the BA1 vaccine?
Yeah, these are also with the BA1 vaccine, but we have been also requested to conduct clinical trial work with the BA4/5 vaccine. And so we are currently in the process of figuring out the best way to do that in the pediatric population. Our strategy all along, as we've been trying to generate data as quickly as possible to enable implementation of a vaccination program before the fall-winter cold and flu season, has been to bracket our investigations of the bivalent vaccine in adults, and particularly in the oldest adults at over age 65, and then in the youngest children, 6 months to 5 years of age, to provide a view of the antibody titers at both of the extremes of age. But we certainly are extending the Kaiser effectiveness study that we've talked about a number of times in this committee together to look at the BA4/5, and that protocol is being extended down to 6 months of age and to booster doses for the pediatric population. So I think that may be the best evidence that we're able to demonstrate in the future.
So, and I'm sorry, I have several questions.
Dr. Sanchez, I'm going to give you one question, and then we're going to go down, and we'll see how much time we have left, okay?
I just want to comment. I'm a little bit concerned that the data that you present in humans is on the, the BA1 vaccine. And, it certainly looks very promising. And I understand the constant shift of these variants, but you have not, these studies with the BA4 and 5 are ongoing in humans. And I'm just wondering whether it's a little bit premature. Thank you.
Yeah, so you're absolutely right that the clinical data that we had prepared for the fall campaign was assuming predominance of the BA1 strain for Omicron. It is why we have looked at the BA4/5 neutralizing capability of the BA1 vaccine, because the two Omicron sublineages are obviously related. So there's about a 4 mutation difference between them, as opposed to about a 35 mutation difference between the original strain and the BA1 Omicron vaccine. But we will continue to generate the BA4/5 containing data. We anticipate that just as the beta containing variant and the BA1 containing variant bivalent data were really consistent with each other. And in the interest of time, I wasn't able to present to you the data from the original beta containing variant cohort, but both in terms of safety and in terms of the patterns of immunogenicity. So the improved immunogenicity to variants of concern, both for the sequence in the vaccine, but also across other variants of concern as compared to the original strain, and the safety data and the reducing reactogenicity profile that we see after subsequent doses has been really consistent between both of the bivalent formulations studied. And so I think by continuing to cover through clinical data the BA4/5, continuing to study, it's really the best we can do to be able to be ready for the fall cold and winter flu season with the vaccine that best matches the current circulating strain.
I understand, and the data that you present certainly look excellent for the BA1 variant. But anyway, okay, thank you.
Thank you. Dr. Paling.
Yes, thank you for this presentation. I wanted to ask about the clinical studies, and if I understood it correct, the longest follow-up at 245 days would be with the beta variant bivalent vaccine. And one of the big questions that we're trying to understand is, to what extent do you see waning? And could you show us some clinical data on the waning with the beta variant over the 245 days? Thank you.
Sure. And while we look for that slide, maybe what I will say is the reason why we have data from the beta variant is obviously because the beta variant emerged in fall of 2020. And so we've had a bit longer to follow those subjects than with the BA1 containing variant, which emerged around Thanksgiving of last year. So let me look and see if I can present to you the slide, though, that you're asking. It's going to take me a minute, and I apologize for that because it's actually CDC that's showing the slides right now. Okay, I found it. Hang on one second. Let me see if I can figure out now on Zoom how to share. Well, unfortunately, I don't think I can because it keeps telling me that host participants has disabled sharing. So I apologize that I'm not able to show you the slide, but perhaps we can provide it to CDC and show it later.
Did you want to describe the slide, Dr. Miller?
Sure, I can do that. I mean, in all cases, the antibody titers with the beta bivalent containing vaccine are numerically higher than what we see with the 1273 vaccine. And the distance between the two actually increases at the 6-month time point. And we believe that there is a biologic basis for this. It's actually why we advocated fairly strongly for the bivalent composition. And it's because when the mRNA in a bivalent formulation is delivered to the cell, both mRNAs, so the mRNA for the original strain spike sequence and for the whatever the bivalent containing sequence is, are delivered to the cell, which means that the ribosomes are translating in the same cell strands of both the original and the variant of concern. These amino acid chains still naturally assemble into trimers, but we've been working with the University of Washington and been able to demonstrate in a publication that we're in the process of submitting that heterotrimers are actually formed. So what that means is, unlike with the original 1273, where the original strain spike sequence is the only one available, we have sequences from both the original strain and the variant of concern. This actually leads to more open conformation and exposure of additional antigens. And we believe that it's exposure to those additional antigens that's leading to the improved antibody persistence, not only against the variant of concern, but against the original strain and other variants as well.
Thank you. And that would be wonderful if you're willing to share that with the committee. We'd really appreciate it. Next, Dr. Cotton.
Thanks very much for your presentation, which was very clear. I was wondering about 3% of the U.S. population is estimated to be immunocompromised, and I was wondering if they were included in any of these cohorts and if you had further data or what plans are for studying that population, which has been exceptionally vulnerable to COVID-19.
Yeah, absolutely. So I didn't present the data today, but we've actually conducted a separate Moderna-sponsored clinical trial in patients with solid organ transplants. And then we've also supported a collaborator in a clinical trial investigating booster doses in patients with solid tumors as well as hematologic cancers. And in both cases, we've been able to observe that the vaccine is immunogenic and does lead to additional immune responses. So in the cancer population, we had studied 3 doses, and then in the transplant population, up to 4 doses. So those data are actually going to form part of the basis of our supplemental BLA for booster dose full licensure as opposed to EUA authorization. And we're actively working on that submission now by the end of the year to prepare ourselves for the 2023 season.
do you think we would have access to that data so that we can better inform our patients as to what the best plan would be for them?
Certainly. The data in cancer patients has already been published, and we'll share that publication with the committee. And then we have the data from the solid organ transplant patients that we can also share with the committee.
Thank you.
Thank you. That would be terrific. Ms. McNally.
Thank you, Dr. Miller. I'm the consumer representative, and I'd like to try and distill down an issue for the public. If you can answer this question, to what extent, if any, did Moderna's research and development process differ in the creation of the bivalent vaccine from the original vaccine? Thank you.
Yeah. So the original vaccine was part of a clinical development program that included the phase 1, 2, and 3 clinical trials. We view the bivalent vaccines as extensions of the SARS-CoV-2 virus. So much like you change your flu vaccine every year as the influenza virus evolves, and you don't study a full efficacy trial each year with influenza vaccines, we are measuring immune responses and inferring effectiveness because we have observed that immune responses really correlate with protection against disease. And those are data that actually Moderna has published in conjunction with the NIH recently in Science magazine, and so, or journal. And so moving forward, as we have done with influenza vaccines, we continue to evaluate the clinical data and compare those immune responses and show the consistency to the original clinical trial.
Thank you very much for the information.
Thank you. Dr. Lehr?
Thank you. I'd like to follow up on Dr. Sanchez's comments and Dr. Miller's last comments. I do recognize that these are animal studies for the B45 variant, but I also agree that this is the future that we're heading for, which is we're going to have more variants, and we should be treating this like the flu, where the variants, if we have evidence that the pattern seems to flow smoothly and that we can use new strain variants every year, I think that's the way we're going to be going over the next several years. So after thinking about it, I am comfortable, even though we don't have human data and just animal data, of supporting the B45 variant booster. Thank you.
Thank you, Dr. Lehr. Dr. Duchin?
Thank you. Related question. The comment that you made about how the mouse study data accurately predicted what we know now through human data for earlier, for the 1273 variant. I'm wondering if you can say a little bit more about that, that would help people understand, the relevance of the mouse data and how it is, at least has been a useful and accurate predictor of what we see in humans.
Sure, Dr. Duchin, and I'm going to start, but then I'm going to ask Dr. Edwards also to fill in because he's much more of an expert in these models than I. But I think the real value of this particular murine model is that the animals express the human ACE2 receptor. And the reason why that's important is the ACE2 receptor in humans is actually the port of entry into cells by this virus, into human cells. And so by using an animal that is expressing that same protein, we're really able to see the impact that the biology of the vaccine has on the animal that would be susceptible to infection in the same way a human would be. And Dr. Edwards, is there anything else you want to say?
Thank you, Dr. Miller. Yes, I think it's highly relevant. Two additional points are highly relevant. One, we now have 2.5 years of experience with these animal models and how they do correlate to human immune responses. And we have seen very good correlation between effective doses and effective bivalent vaccines between mice, non-human primates, and humans to this point. One particularly relevant point, though, is we do measure in these animal studies the impact of variants on neutralizing titers. So, for example, a Ba1 neutralizing titer in a mouse vaccinated with 1273 is many-fold reduced versus the original strain. And we see the same thing recapitulated when we're assessing human sera. So both from a immunogenicity standpoint and an impact of variant standpoint, these animal models have translated very well.
Those are very helpful comments. Thank you.
Thank you. Dr. Sanchez?
Thanks again. Question, studies in pregnancy, and then the other one is really more basic and just trying to get an idea of how the vaccine is formulated. Are these two different messenger RNA strains that are encoding the different spike proteins, or is this one strand that encodes both? And does the ultimate protein cross the placenta?
Okay, again, I'm going to start, and then I'm going to actually ask some other experts to help me, particularly Dr. Edwards. But maybe first to start with respect to pregnancy. So we are conducting a safety follow-up study in pregnant women. It's a registry where the study is currently ongoing and will examine approximately 800 pregnancies overall. The question around the mRNA sequences. So there are two distinct mRNA sequences. The first sequence is the original sequence that was in mRNA 1273. It encodes for the full-length spike protein from the original Wuhan strain. The second sequence includes the sequence from a BA.4/5, and it's important to note that the spike protein sequence is identical from BA.4, BA.5, which is why we refer to it as a BA.4/5 sequence. Those two are individual sequences on lipid nanoparticles. Those lipid nanoparticles, though more than one, is able to enter the cell, and that's how both mRNA sequences are able to be translated inside the same cell. And then in terms of transfer across the placenta, so we have conducted developmental and reproductive toxicology studies with mRNA 1273, but also with other vaccines in our pipeline, maybe in particular a CMV vaccine that's actually a hexavalent vaccine. And we do not see that the pregnancy or fetus is impacted by vaccination. But I'm going to ask Dr. Edwards to comment further on the pregnancy piece.
Thank you, Dr. Miller. So on the mRNA response, just one clarification. So the two mRNAs that are included are co-formulated in the same lipid nanoparticles and delivered then to the same cells. Further, we also introduced to both mRNAs the two proline mutations that stabilize the conformation of the spike protein into the prefusion conformation. And then on the pregnancy piece, we do have evidence from animal studies that there is placental transfer of both IgG and to a limited degree IgA, and that also includes maternal transfer via breast milk.
Thank you.
Protein. Sorry.
What was your question?
How about the protein that's the protein that's generated by the messenger RNA? Does that cross the placenta?
Thank you for that. So we have done developmental and reproductive toxicity studies. And maybe we can come back at a later time point during this call to share the results from those studies. I unfortunately am not the expert on that study, and I don't believe our tox expert is available. But we can get back to you later on this call. Thank you.
And Dr. Edwards, I apologize. I was on mute. What I was going to say, Dr. Sanchez, is I didn't understand your question initially, but I mean, in terms of the protein, this protein has been engineered to be cell surface expressed. So it's not a protein that is secreted in the same way that like a subunit protein might be injected and flowing freely. It is mRNA that is entering the cells, and then the protein itself is cell surface expressed. So it's not secreted protein.
Thank you.
Thank you. I have one last question, then we should move on, and hopefully mine is quick. On slide 7, I was noting the interval between the 3rd and the 4th dose. Looks like the average is 4.5 months, but the lower range is at around 3 months. And so this is probably a combination question. The authors to Moderna and to FDA, but the lower band or sort of lower end of the approval is 2 months from the last prior dose or the last dose. Do we have any safety data for that 2 to 3 month window?
So we are generating data with respect to the 2 month window in collaboration with the NIH in the COVAIL study. The data we have available now comes from previous collaborations with the NIH in the 3 month window. And I think the 2 month window had been requested of us based on the desire to make boosters available prior to potential increases in infection rates. But others may want to comment, perhaps from the FDA.
Hi, this is Doran Fink from the US FDA. So that's correct. We don't have clinical trial data or significant real world data with a booster dose at a 2 month interval. However, we do have data with booster dose intervals ranging from 3 months through up to 6, 7, 8 months or longer that for vaccine reactogenicity really don't seem to show a difference in the level of reactogenicity compared to the interval. I know that one specific concern about the interval is the risk of vaccine-associated myocarditis that has been observed most prominently following the second primary series dose and also a first booster dose in certain populations. We don't have data one way or another that would suggest that the risk of myocarditis would be higher or lower following an interval of 2 months as opposed to, say, 3 months. And frankly, any clinical trials of the size that we really are able to look at for considering the authorization of these booster doses wouldn't be adequately powered to look at myocarditis anyway. We do have experience, as the committee knows, with the risk of myocarditis relative to the primary series interval between first and second doses, and those data indicate that the risk when the primary series doses are separated by at least 2 months appears to be lower than when the primary series doses are administered closer together, and no further reduced risk with intervals longer than 2 months. So based on the totality of evidence that we do have at hand, and also considering the timeliness of making this booster dose available to individuals who may have received their last vaccination more recently, those considerations really underlie FDA's decision to authorize these bivalent boosters with a minimum interval of at least 2 months. I also do want to stress that we understand that most people who would be eligible for these second boosters will have received their last COVID-19 vaccination well beyond 2 months previously, and so we are talking about really a relatively small fraction of individuals who might be considering getting one of these bivalent boosters at an interval of close to 2 months. Thank you.
Thank you both for that thorough response. One last question for you from prior. I just want to make sure we close the loop. The question came up at the last meeting and earlier during this meeting around the VAERS adverse event reporting regarding potential administration errors and the concern that the committee has had with regard to labeling. could you comment on whether, you commented on the, hopefully single-dose syringes, but is there any plan to modify or make that labeling more clear to minimize the risk of administration errors going forward?
Yeah, I'm going to ask my colleague Luis from the manufacturing group to maybe comment further, but it certainly is the intent that we get to product-specific presentation for each individual presentation. But Luis, anything else you want to say?
Yeah, good afternoon. This is Luis Mustafa, VP of CMC Strategic Operations. Yeah, Dr. Miller, you're absolutely correct. It is in our plan or in our forecast to move to a specific presentation by age. As we move out of the pandemic configuration, we have that as part of our long-range strategic planning for having a specific SKUs, a presentation for them.
Okay, thank you. I mean, it would be really very much appreciated to sort of have a timeline and a sense of when that will occur. In the interim, I do feel like some of the pediatric vaccination programs
Dr. Lee, I think you're muted.
We can't hear you, Dr.
The
Lee.
next presentation. Thank you, everyone.
We didn't hear you, Grace.
You cannot hear me?
Or you cut off for the last.
Oh, no.
Yeah.
My request was to actually both manufacturers, either to FDA or to CDC to provide a clear plan and timing of that plan for when changes would occur to the labeling. Because I just honestly feel terrible about the fact that there are so many administration errors that seem disproportionate to what we've seen with other vaccines or with the adult vaccines. And I feel pretty strongly that anything we can do to support implementation by providers and by public health and by pharmacists, it would be extremely helpful in the delivery of these vaccines and getting them to kids that need them. So that's my request out to both companies. But let's move on to the next presentation because we are running out of time. So I would like to move on to the next presentation by our colleagues from Pfizer, and who will speak about the Pfizer-BioNTech COVID-19 Omicron-Modified bivalent vaccine candidate. And it looks like Dr. Swanson will be presenting. Please go ahead, Dr. Swanson.
Great. Thank you very much, and good afternoon, everyone. My name is Tina Swanson, and I am the head of viral vaccines R&D at Pfizer. And on behalf of Pfizer and BioNTech, it is my pleasure to share both immunogenicity and safety data today for our bivalent Omicron-modified variant vaccine. So if we could go to the next slide, and then I think we can just go straight to the next one. So this is likely a familiar graphic, but really we want to highlight here that, of course, throughout the pandemic, we have seen the rapidly changing SARS-CoV-2 variant epidemiology indicated by each of the colors on this graphic over time. And in particular, the emergence of the more antigenically distinct Omicron variant of concern, with demonstrated increased transmissibility and evidence for partial immune escape. So this brings us to the focus of today's discussion on the bivalent vaccine that includes an Omicron BA4, BA5 component to address COVID-19 due not only to the Omicron sublineages, but also due to potential subsequent variants of concern. Next slide. So over the past 2+ years, we have gained substantial clinical experience with variant-modified vaccines across different age groups, and this is illustrated on the present slide. We initially evaluated early on in the pandemic when the Beta variant of concern was of initial particular interest when it initially emerged. We evaluated a monovalent form of the Beta variant modified vaccine in individuals 18 to 55 years of age, both as a primary series and as a third and fourth dose booster. And of course, more recently, we have subsequently evaluated the Omicron BA1 variant modified vaccine, both as a monovalent formulation in 18 to 55-year-olds as primary series and fourth dose booster and a bivalent formulation in greater than 55 years of age. And those data will be in today's presentation. And we do also have ongoing study of the bivalent BA1 vaccine in 18 to 55-year-olds. And throughout the clinical evaluation of each of these vaccines, the preclinical data also generated with these same formulations have reliably predicted responses in humans. And so that's across the beta monovalent, the Omicron BA1, both monovalent and bivalent. And of course, now the focus of today's discussion being the Omicron BA.4/5 bivalent vaccine. We have an ongoing clinical study evaluating this vaccine in individuals from 12 years of age to greater than 55 years of age. And we'll also describe some of the preclinical data, which again has shown some evidence that we anticipate will be translating to observations in humans, as we've seen in the prior preclinical evaluation of variant-modified vaccines. Next slide. So first, to go through the immunogenicity data with our Omicron BA1 containing variant vaccines. So I want to show a couple of data slides around actually the monovalent composition, and this supports some of the discussion of a bivalent vaccine approach. So in this first clinical study, which included participants 18 to 55 years of age, we evaluated a primary two-dose series of the monovalent Omicron BA1 containing vaccine at a 30 microgram dose level. And if we go to the next slide, we can show the neutralization responses in a sentinel cohort of these individuals. So these are participants without evidence of prior SARS-CoV-2 infection. And these are the serum neutralizing titers 1 month after the second dose of a primary series of the monovalent Omicron BA1 vaccine. And if you focus on the dark blue bars on the far right, you can see the Omicron BA1 neutralizing titers in dark blue, and Delta in the middle in green, and the reference strain USA Washington 2020 in gray on the left. And what we observed is a very Omicron-specific neutralizing response with a monovalent Omicron BA1 vaccine in naive individuals. So if we go to the next slide, now we're looking at preclinical data. We can go to the next slide. There we go. So in the preclinical study, if you focus on the middle of this figure, it's the same monovalent Omicron BA1 containing vaccine, again evaluated at the same dose regimen, so days 0 and 21 in naive mice. And this was at a dose level of 0.5 micrograms. You see again a very Omicron-specific response with the BA1 strain in dark blue and the BA.4/5 in light blue. And in addition, we also evaluated neutralization against not just the reference strain, but also Beta and Delta. And then if you focus on the right-hand side of this figure, you can see the bivalent composition. So the original ancestral strain spike combined with the Omicron BA1 spike, which elicits a more balanced immune response across the different variants of concern, including the reference strain in this preclinical study. So providing some evidence and support that bivalent will provide a better and broader immune response compared to a monovalent approach in a naive background. Next slide. All right, so this gets into the evaluation of the bivalent Omicron BA1 containing vaccine. And in this clinical study, this was in participants greater than 55 years of age. And the either bivalent Omicron BA1 containing vaccine at a 30 microgram dose level was administered as a fourth dose, or the prototype vaccine at 30 micrograms was administered as a fourth dose. And that fourth dose was administered a median of 6.3 months from dose 3. We go to the next slide. So first, we wanted to understand the Omicron BA1 neutralizing antibody response as part of the evaluation of the superiority criteria that needed to be met in order to demonstrate a substantial improvement in the Omicron BA1 neutralizing response with the bivalent vaccine compared to the prototype. So you're seeing in this table the geometric mean neutralizing titers of Omicron BA1 neutralization activity, and this is in the validated SARS-CoV-2 neutralization assay. And in order to meet superiority criteria, the geometric mean ratio of the neutralizing response in the bivalent vaccine had to be greater than 1.0 to meet simple superiority criteria. And this is for the lower bound of the 95% confidence interval. So if you see the yellow box on the far right, you can see that superiority was met with a lower bound of 1.17 and a GMR of 1.56. We move to the next slide. So this is now looking also at the Omicron BA1 neutralization response, but instead of GMR, evaluating seroresponse rate. And for this particular analysis of seroresponse, non-inferiority criteria needed to be met with a lower bound of the 95% confidence interval for the percentage difference in the seroresponse rate between the bivalent vaccine group and the prototype vaccine group being greater than -5. You can see the lower bound of the confidence interval for the bivalent vaccine was greater than -5, which had a lower bound of 4.0. So non-inferiority met for the seroresponse rate. Next slide. And this is just to show the both before vaccination and 1 month post-dose, the substantial increases observed in Omicron BA1 neutralization activity in the bivalent vaccine containing group compared to the prototype. So the geometric mean fold rise from before vaccination and 1 month post fourth dose is shown across the top in the yellow box with 9.1 for the bivalent vaccine compared to 5.8 for the prototype. Next slide. So moving beyond the Omicron BA1 neutralizing response, it was also important in this bivalent composition, which also includes the original ancestral spike, to demonstrate non-inferiority of the reference strain neutralizing response. So here you're seeing, again, in a validated SARS-CoV-2 neutralization assay, the results of this analysis. And in this case, for non-inferiority criterion, the lower bound of the 95% confidence interval for the geometric mean ratio between the bivalent and the prototype vaccine required to be greater than 0.67. And you can see we had a GMR of 0.99 with a lower bound of 0.82. So non-inferiority criterion was met. Next slide. Okay, so that is the summary of the immunogenicity data. And now we wanted to share a summary of the safety data. So what you're seeing on this slide is a summary of the reactogenicity profile of the bivalent Omicron BA1 containing vaccine given as a fourth dose compared to the prototype vaccine given as a fourth dose in this same study in participants greater than 55 years of age. And I've just highlighted in green in both local reaction at the injection site focusing on pain as an example, and then in the systemic events category highlighting fever. You can see we have a very similar reactogenicity profile between the variant modified vaccine compared to the prototype vaccine. And not shown on this slide, but we also have data we can share later showing that we've seen similar trends in the original beta variant modified vaccine study and also with the monovalent Omicron BA1 containing vaccine. Next slide. Okay. So of course, we've seen over time the emergence of the BA4 and then now the dominance of BA5. These two sublineages encode the same spike antigen. So we evaluated the neutralization activity both with the prototype vaccine group receiving the fourth dose and the Omicron BA1 containing vaccine with the fourth dose. And we see nice increase in the Omicron neutralizing response in this subset analysis. We do see neutralization of BA4/5. However, you can see this is reduced compared to the BA1, which is why we have subsequently gone on to evaluate a BA4/5 containing bivalent vaccine. Next slide. So before getting into some of the preclinical data on the BA.4/5 variant modified vaccine, this is just to round out the translation of preclinical to clinical data. So similarly, when we showed the primary series analysis in mice translating to observations in humans, this is looking at the booster setting of the Omicron BA.1, both monovalent and bivalent, in mice that have received 2 prior doses of the original BNT162b2 vaccine. And you can see with the monovalent again in the middle and the bivalent on the right-hand side and the prototype vaccine on the far left, we see similar trends with improved Omicron neutralizing responses in the Omicron-containing vaccine groups, but similarly reduced neutralization activity against the BA.4/5. So a nice translation of preclinical to clinical data. Next slide. All right, so this gets into the BA.4/5 monovalent and bivalent vaccine compositions, which were evaluated as a booster dose, again, looking at a 3rd dose in mice that had previously received 2 doses of the BNT162b2 vaccine. And so the neutralization data that you're seeing on this slide is 7 days post 3rd dose, and evaluating neutralization activity against the ancestral strain, in this case, which is Wuhan Hu1, compared to as well the Omicron sublineages BA.1, BA.2, 212.1, and BA.4/5. And the key takeaway for this slide is the 2 groups on the far right, which is the monovalent Omicron BA.4/5 in pink and the Omicron 4/5 bivalent in purple on the far right. You can see a much more balanced immune response against the Omicron sublineages with the 4/5 containing vaccine, including substantial increases, of course, in the matched strains of BA.4/5 compared to the Omicron BA.1 monovalent and the prototype vaccine. So following this initial evaluation in mice, we subsequently performed a follow-on study to confirm these results, and that's shown on the next slide. So here again is looking at the monovalent BA.4/5 vaccine on the left and the bivalent 4/5 vaccine on the right. And again, you see a nice balanced response across the Omicron sublineages when these vaccines are administered as a 3rd dose booster in mice. We can go to the next slide. So overall, we've shown, number one, of course, the reactogenicity profile of the variant vaccines that have been evaluated clinically, which include Beta, Omicron BA.1. We have also evaluated Alpha and Delta in prior clinical studies. And in each of these studies, the reactogenicity profile has been similar to the prototype vaccine, BNT162b2. In the evaluation of the Omicron BA.1 containing vaccines, both monovalent and bivalent, we demonstrated superiority for the Omicron BA.1 geometric mean ratio and non-inferiority for the seroresponse, as well as non-inferiority for the reference strain geometric mean ratio. And across the evaluation of variant-modified vaccines to date, the preclinical immunogenicity data have reliably predicted observations in humans. And including the latest Omicron BA.4/5 modified variant vaccine booster data in mice, supporting improved neutralizing responses across Omicron sublineages. So collectively, we anticipate to see similar trends in the ongoing BA.4/5 bivalent clinical study. So to summarize, the emergency use authorization has been granted for use as a booster dose for eligible populations 12 years and older, the bivalent Omicron BA.4/5 variant modified vaccine at the 30 microgram dose level. And just to wrap up the presentation, just to note that we will also be further evaluating clinically the 4/5 bivalent vaccine in pediatric populations. Next slide. just to briefly thank the committee and of course the participants in our clinical studies, the investigators, and my fellow colleagues at Pfizer and BioNTech, and I would be happy to take any questions.
Thank you, Dr. Swanson. This presentation is now open for questions. Ms. McNally.
Hi, Dr. Swanson. I am the consumer representative. I wanted to ask you the same question that I asked Dr. Miller regarding to what extent, if any, Pfizer's research and development process differed in the creation of this vaccine from the original vaccine. Thank you.
Right. So I will probably provide you a very similar response. So I think what's important to note that from the very first variant modified vaccine that we evaluated, which was the beta, monovalent, through the current Omicron BA.4/5, the only change is the variant-specific sequence changes in the mRNA itself. All of the processes from both making the mRNA drug substance to formulating into the lipid nanoparticle follow the exact same process as has been used throughout generation of the COVID-19 vaccine, BNT162b2.
Very helpful. Thank you.
Thank you. Dr. Paley? Dr. Paley?
Okay, can you hear me now?
I can't. Oh, there we go. Yep, thank you.
Oh, apologize. I too wanted to ask you a very similar question that I asked Dr. Miller. And among your variant studies in humans, do you have any long-term follow-up, and what have you learned about the persistence or the waning of the immune response? Thank you.
So we do have ongoing follow-up for looking at the duration of the antibody response in our clinical studies. We're still, following out to later time points, 6 months and beyond. The data that we currently have available on persistence would be in a subset of participants that received 3 doses of the BNT162b2 prototype vaccine, and we evaluated activity against the reference strain and the Omicron BA1 variant of concern at the time. So this was before BA.4/5 came along. And we see similar antibody decay rates between the reference strain and Omicron BA1, at least out to 4 months after dose 3. So we're continuing to follow with more subjects and more time the overall kinetics, but those are the data that we currently have available.
Thank you. Dr. Cotton?
Thank you for your presentation. Similar to my last question, for immunocompromised patients, what work is underway and what information might inform our decision-making for this vulnerable population?
Yes. So maybe I can make a start, and if there's anything else to add, I'll ask Dr. Nick Kitchen to add any further comments. So we are conducting a study in immunocompromised individuals, but to date, the enrollment rate has not been, as expected, since the vaccination in this population is generally recommended. So at this point, we don't yet have data specific to that study in the immunocompromised group, but maybe I'll ask Nick Kitchen to see if he has anything else he would like to add.
Thanks, Keenan. No, I think you've summarized well. Obviously, with broad recommendations in most countries now for immunocompromised to be vaccinated in the face of the threat of SARS-CoV-2, it has been, as you said, difficult to enroll individuals into clinical trials. So we don't have data yet in that population.
What about research looking at these additional doses in people who had the appropriate number of vaccines given already? Has anything been done there, or do you have any plans for that?
You mean administration of the bivalent BA.4/5 vaccine to immunocompromised individuals? We don't currently have a study ongoing in that regard, no.
Okay, that makes it challenging for us to think about how we would best couch our recommendations. So I would encourage you to think about studying that highly vulnerable population. Thank you.
Thank you.
Thank you. Dr. Sanchez?
Thank you. And so my comments would be similar, and I presume the packaging of this vaccine is similar to what was described previously by the Moderna. But my question again is your studies on pregnancy. And then I have a question also on slide 17. So, I know you presented the data on the BA.1 vaccine that I guess my idea when different, combination vaccines, does it affect the immunogenicity of one component versus the other? And I know with your BA.1, certainly it did not appear, it was not affected. But when I look at this one, and I think in a previous slide also in the mice, the levels of, the neutralization titers in some of these look lower than with the combined BA.4/5 variant than with the one alone. Those may not be significant, but can you comment on that? And I think it was also in the previous slide that you also had looking at it with in the mice as well. And I guess my concern would be if they generate lower antibody neutralization titers, would we expect the decay to be faster? I guess I have several questions.
Maybe I'll ask one clarification. Are you referring to monovalent versus bivalent presentation of the variant vaccine?
Yes.
Okay. So in the clinical study, we did evaluate in individuals greater than 55 years of age, monovalent Omicron BA.1 versus bivalent Omicron BA.1. And we did see a trend for slightly higher Omicron BA.1 neutralizing responses in the monovalent vaccine group compared to the bivalent group. Whether that difference is a clinically meaningful difference, I don't think we can speculate. But what I can say is that preclinically, we are seeing similar trends with slightly higher strain-matched responses with monovalent versus bivalent presentation, both with the BA.1 containing and the BA.4/5 containing vaccines. And I think I would also refer back to the primary series immunogenicity data that showed clear benefit of having that bivalent composition. So I think what the goal here is to ensure that we're solving for the current circulating variants, but also anticipating potential future variants by applying that bivalent approach. So I don't know if that answers your question, but I think that's the general concept.
No, I understand. But when you look at this slide here and the monovalent BA.4/5 versus when you combine the two, is that a statistically significant decrease in titer or not? I guess that's my question.
Yeah, I don't think so. I think the confidence intervals, it's probably not shown clearly on this slide, but we can run, I can go back and look at the analysis and follow up with you, but I think it is overlapping.
Okay. And pregnancy?
So we do have a study that we have ongoing within pregnant women. And I think we also have run into similar challenges of enrollment, given that the vaccine was made available for this group. But maybe I'll ask Nick Kitchen to, again, if he would like to add any more details to that.
Yeah, thanks, Kina. Yes, indeed, that's correct. We did conduct a study in pregnant women for which the data analysis is ongoing with following not just the pregnant women but also their infants as well. So that was with the original vaccine, but we will be generating data from that study despite the difficulties in enrollment, again for similar reasons due to the wide recommendations for pregnant women to be vaccinated, but we will generate those data for sure.
Thank you. Dr. Daly.
Yeah, thanks. Because of the concerning data presented earlier about vaccine administration errors, I want to ask that question. I realize we're focused primarily on the adult presentation here, but what are your specific plans around presentation of any pediatric products to minimize dose administration errors, and kind of what's the timeline for that plan as well? Thank you.
Thank you for the question, and I'm also recalling that there was a question on also the current rollout of the BA.4/5 for the 12 years and older. And so maybe if we could bring up the last backup slide in the deck, which should be slide 37. And while that's coming up, I'll ask Adam if he can speak to first the BA.4/5 presentation and then also the pediatric files.
Thank you, Kina. Can you hear me okay?
Yes, I can. Thank you.
Okay, brilliant. If we could go to the next slide, please, just to cover the first question related to the bivalent 30 microgram dose. I think this may be a slide build. If we go to slide 38, please. Thank you very much. So the Omicron-containing bivalent vaccine will retain the gray cap as per the original vaccine. And the reason for this is the Omicron-containing bivalent vaccine is essentially the same drug product. It contains the same formulation as the original vaccine, albeit it contains the different strain, much like the flu vaccine. And the gray cap is a visual indication that the product is the same dose, requires the same storage conditions, and the same handling as the original cap presentation. And you can see from the information on the right-hand side of the slide is that we differentiate between the original primary series vaccine, so the original vaccine, and the BA.4/5 containing bivalent vaccine through the label that is highlighted on the right-hand side. So I'll pause there. Does that answer the question related to the gray cap presentation, which is the bivalent BA.4/5 containing vaccine?
Yes.
Yeah. Well, Dr. Daly, if you don't mind, I'll just jump in and say, maybe when we get to the clinical considerations, it'll be clearer why we're asking these questions. But I do think that, dilution, no dilution, colors of the caps, all of these things have just been a bit overwhelming. And actually, I still don't see on here like the microgram dosing, so that is it 30/30 or is it 15/15 for the Omicron components?
15 and 15.
15/15, and like having that information on the dosing would be incredibly helpful. Hopefully, we're beyond the point where the expiration date is actually accurate and all the information on the label is actually accurate. Is that correct now?
That is correct.
All the pediatric formulations?
That is correct, yes. And just to make a quick statement around that, this I think it's been flagged as a potential mislabeling, which is not the case. We were labeling the product conservatively with the information that was available at the time to try and make sure that we had the product available to patients as soon as possible. And as soon as we got the information to update the label and the approval that allowed us to update the label, that was done so as quickly as possible.
Okay, thank you. So maybe we can revisit this. I just want to make sure that we are clear in our recommendations to our own providers or our own health systems about what the right thing to do is. But again, if there's any questions or continued confusion, we'd appreciate your partnership on that, and also any transparency you have on any updates. Let's go to Dr. Paling, and then we'll go to Dr. Deeks and Dr. Anderson.
I'm sorry, I forgot to take down my hand. Thank you.
Thank you. Dr. Deek?
Thanks very much. I just have a question for a clarification. In terms of your slide with respect to your various studies, it looks like the monovalent beta and Omicron went down to age 18. And of course, the bivalent studies went to 55 years, over 55 years of age. So can you clarify that the bivalent BA.4/5 has actually not been studied in anyone 12 to 17 years of age, at the age group for whom there is an authorization? Thank you.
So thanks for the question. I think if we could go to slide 4. Okay. So you're correct, the bivalent Omicron BA.1, the data that was presented today and that we currently have available is in individuals greater than 55 years of age. Within that same study, we've also enrolled and will be generating additional data in participants 18 to 55 years of age with the BA.1 bivalent containing vaccine. For the BA.4/5, you can see the age groups listed here. So we've actually stratified the 4/5 clinical study to ensure we have sufficient numbers of individuals 12 to 17 years of age, 18 to 55 years of age, and those greater than 55 for receiving the BA.4/5 bivalent as a fourth dose booster. So if there are more specific questions that I can ask Nick Kitchen to address them, but if that answers your question, let me know.
Yes, it does. So just no data in humans yet for 12 to 17. Thank you.
Study is ongoing, correct.
Thank you. And Dr. Anderson?
Yeah, so this is Dr. Anderson from Pediatric Infectious Disease Society. Dr. Deeks actually asked the question that I had planned to ask. But I did want to say thank you to both manufacturers for what sounds like efforts to move clinical trials of the bivalent vaccines into children of all ages. And obviously, we anxiously await all that data. Thank you.
Oh, thank you. And Dr. Long, last question.
Yes, I want to thank you also, both manufacturers, for moving so quickly and bringing this data. And now, my problem is a little bit, there aren't many data upon which we really can make confident decisions. Do I understand? Let's just talk about your bivalent and the bivalent for BA.4 and 5. You don't have any human antibody decay, even over, or response, even over a very short period of time. I guess my thought is, and it's really naivete on my part, we've seen that many doses have been purported to lengthen the antibody response. But we haven't seen any data on whether you add something new, which is as distant as BA.4 and 5, and are you still going to have to see that 3 and 4 times before there's more than 3 months? So as we just decide on whom this vaccine might be used, I just want to be clear that we're seeing all of the clinical data, all the human data that you have on 4 and 5. Which is little.
Thank you, Dr. Long. This is a very good question. I think that where I would, focus on is, yes, we have extensive data, of course, on the prototype vaccine, where we have seen very consistent responses through the 3rd dose against both the ancestral strain and different variants of concern, including Omicron. And with the BA.4/5 bivalent, that study is ongoing, so we don't yet have specific clinical data for that bivalent vaccine composition. However, the Omicron lineage has been the most antigenically distant variant of concerns to date for SARS-CoV-2 throughout the SARS-CoV-2 pandemic. And when you combine a much more antigenically distinct spike with the original ancestral spike, the more distant there is, just the following of the diversity of antigen-specific memory B cells over time, the anticipation would be that combining something so distant, you would have some chance of improving upon not only memory B cells recognizing epitopes that are shared between the ancestral and the BA.4/5, but also potentially specific to the BA.4/5. So a broadening of that protective immunity. And of course, we'll generate the data, but that, it is within the realm, I think, across different disease areas, including, for example, influenza, where that could be an anticipation in the data that we will see. We have also seen in the preclinical studies a maturation of the neutralizing antibody response. So the initial data with a third dose booster of the BA.4/5, the data in this presentation we had available to include is 7-day post dose 3. But we've also gone on to look at 1 month with the BA.1 containing bivalent vaccine and now generating data for the BA.4/5. And we do see nice continual increases in the neutralizing antibody response against either the BA.1 or the BA.4/5 over time. So I think there is some opportunity here that we may see some expansion in the breadth of protection.
Thank you very much. I just want to ask, do you have an anticipation of when you might be asking for authorization for boosters for the bivalent in the 5 to 11?
So maybe I will ask, I think we have Bill Gruber with us as well today. So Bill, would you like to speak to that?
Yeah, I'm happy to answer that, Dr. Long. We anticipate that we'll be taking data from the existing study, the BNT162b2, in the 5 to 11, that booster data, providing information about cross-active immune response against BA.1, BA.4/5, and anticipate we would file for submission sometime in the first part of October. And then, as Keena said, just maybe anticipating an additional question, we obviously, as Keena said, are working with our FDA partners to identify the best way to move forward into the younger age groups with appropriate trials and getting data as quickly as possible so that we could further extend the potential for the bivalent BA.4/5 and original containing vaccines as soon as possible.
Thank you.
Thank you. And with that, I'd like for us to thank our speakers today and for all the questions and clarifications that were given. And we will end the session now. Why don't we reconvene in, I'm actually going to give us a whole 15 minutes today. So we will come back at 45 minutes after the hour as planned to return to Dr. Oliver and Dr. Hall to hopefully wrap us up. Thank you, everyone.